Good morning everyone. Myself Manpreet and my partner Harman going to discuss our research topic isolation and dereplication of actinomycete isolated from natural sources. And the natural sources we are using here, number one is the soil. We have taken samples from various soil. The second is the marine one, and third one is our plant-based sources. Now in this report I'm going to show you the contents we are going to discuss. And for the content first I will introduce the topic, then our material and methods, techniques, results, discussion and conclusion. If we talk about the abstract, so in the abstract, why we are doing this research, so our research will provide us the process of isolation and identification of actinomycetes from various natural environments, which include, as I earlier mentioned, soil, sediments and ecosystem. Why we are doing research? So that we can find new compounds, which is the well-known capacity of actinomycetes to create a wide range of secondary metabolites with substantial medicinal promise. And secondly, we are using the dereplication techniques so that we are able to identify and isolate new strains if present in the various natural sources. So the dereplication procedures we are using is molecular fingerprinting or bioactive profiling. And why we are using this technique? So that we are able to find new bioactive compounds which will be useful in our medicinal purposes. Next, introduction. So in the introduction, we are going to discuss that what is actinomycetes. Actually, actinomycetes is a genus which is having capacity to produce a wide range of secondary metabolites, so that it can transform into medicinal valuable antibiotics, anti-cancer drugs and immunosuppressive compounds. Although there are a number of actinomycetes derived compounds which have been identified by continuous process of finding new molecules, it means we have having wide number of area where we can explore more. So the extraction of actinomycetes from the natural sources are very crucial stages, are the crucial stages in the drug development. So now I am going to discuss about the material and methods we have used for this research. So the number one is our isolation techniques. So before discussing the isolation techniques, I would like to show the electron micrograph. So this is the cross section of actinomycetes. So at the arrow which is indicating is the filamentous of the actinomycetes. So it is actually 0.22 micrometer, and this is the scanning electron micrograph. Now to discuss our material and methods. So the first technique is how we have isolated actinomycetes from the soil specimen. So we have taken the soil especially from 5 to 15 centimeter of surface. Why we have doing this? Because so that we can get a wide range of microbial species in there. So after that we have dried the soil for 48 hours. Then we have done the dilution, so that we are able to get a particular concentration. Then these diluted samples were then spreaded on actino isolation agar, and which is having some kind of antibiotics in it, because we need to prevent the growth of the fungi. After that, the plates were incubated at 28 degrees for the maximum duration of 4 weeks. Then we are able to see the growth of the actinomycetes. So now I am going to show you the various plates. So these plates are basically showing the soil sample but on different media. So as we have used 4 different media, that is Benedict agar, yeast agar, asparagine dextrose agar and Bennett agar. Next is our isolation from marine sample. So same, we have taken the sample from coastal location by 5 to 10 centimeter below the surface, and then the samples were treated with the sodium chloride solution so that it can replicate as same as in the marine conditions. Then the sample is again isolated. Then we have spread it on the MI agar a medium which is having higher concentration of NaCl, and we have also used the antibiotics like in the soil we have used, so that we can prevent the gram-negative bacteria. Then the plates were incubated at 28 degrees Celsius to 2 to 6 weeks in the presence of oxygen. Then we will able to see the characteristic of actinomycetes with the purified colonies. Next is our plant associated sample. So in this we have used certain sterilization techniques. So the samples were firstly disinfected so that we are able to get pure colonies. The disinfected samples were placed on an AIA media mixed with the nystatin. So here also we have used antibiotics so that we can avoid the fungal growth, because the fungus and actinomycete almost require the same temperature and all the environmental conditions to get pure isolates. The emerging colonies will form filament and mycelium like fungus form, and then we will transfer it to the fresh media.
So now I will continue with dereplication strategies. So what is dereplication? Why we are using this strategy? Because it is very important process for the identification of new bioactive compounds which are natural products and which are extracted from natural product, and especially these are dealing with microorganisms and actinomycetes, and these are renowned for their abundant synthesis of their secondary metabolites. So this approach is very useful to detect the compounds at the early stage of the exploration. Now we will discuss about its molecular techniques, that how we will identify it with the help of molecular techniques. So we will identify it with the 16S RNA gene sequencing technique. In this approach offers a strong and reliable structure for quickly identifying and categorizing the actinomycete isolates, namely with the level of genus or species. So 16S RNA gene has areas that are highly conserved, and these are used to building the universal primers. So these are variable regions that give specificity to different taxa, and this process of the sequencing of specific gene fragment, and then we are comparing it with the NCBI database gene bank that help to distinguish between already identified actinomycete strains and those are new or which are unknown. So this technique is crucial for determining the order of importance of isolates for subsequent chemical analysis. So now we will move to the chemical screening, that how we can identify with the help of chemical methods. So we do have a lot of methods like HPLC, NMR, MS. So in this we are using MS, which is highly advantageous in the detection and identification of molecules by analyzing their mass to charge ratio. Hence it offers a comprehensive insight into the molecular weight and structure of substances. We are using NMR spectroscopy also because it enhances and process by providing valuable information about the structural categorizes of molecules and such as their functional group, molecular framework. By utilizing these all techniques in combination, it becomes possible to quickly identify and recognize the chemicals present in the actinomycete extract, and these can be simplifying the process of bioactive molecules. So now we will discuss about the results which we got from the isolation methods and dereplication techniques. So after all the methods which we used for the isolation, we got to the result that total 150 actinomycete strains were isolated from the soil samples, which usually contain the genera Streptomyces, Micromonospora and Actinoplanes. These were forest and agricultural soils which were abundant with the organic matter. That's why we got that much of actinomycete isolates. After that, total 75 isolates were obtained from the maritime settings, which are usually belong to the Selenispora and Nocardiosporis genera. So after that, I got 50 isolates which were obtained from the plant materials and usually from endophytic actinomycetes, which belongs to Streptomyces and Micromonospora genera. So these isolates were primarily obtained from the medicinal plants, suggesting that the potential symbiotic connections that contributes to the bioactivity of plant derived medications. So now I am going to discuss about the graphical representation of the different actinomycetes isolates. So we can see that this graph shows the volume-wise identification of actinomycete species, where the large circle shows the Micromonospora. It represents that the higher identification in the sample is followed by the medium circle Nocardia and then the small circle Streptomyces. So now we are going to discuss about dereplication strategies. So in the dereplication process, as I earlier discussed, that we use 16S rRNA gene sequencing method, and after that the chemical HPLC, MS and NMR spectroscopy method. So when we did 16S rRNA gene sequencing, we got 50 potential candidates with distinct GNA species. So after that we used chemical methods, NMR and HPLC, and from all these 50 profiles we got 25 different strains. Then we matched its characteristics with current database, which suggests that there are some new metabolites which were 10 recognized possible creators of innovative substances, which were mostly obtained from the soil and marine samples. So these were distinguished by the distinct spectrum data that suggests the presence of new molecular structures. So this diagram represents the gel and the phylogenetic tree of 16S rDNA sequencing that is presented, along with the matching location and the site of isolation as well as their molecular groups. So for the discussion, we can say that the soil environment in particular tend to provide a greater method of isolates due to its abundant microbial diversity. This abundance in the terrestrial settings frequently surpasses the yield obtained from the marine and plant associated habitats. Nevertheless, the two mentioned sources are primarily noteworthy due to the provision of distinct, less documented variations. This indicates the urgent requirement to expand research endeavors across diverse environments in order to fully explore the range of microbial diversity. They contend the incorporation of several scientific fields that can greatly improve the detection and creation of new substances, especially from unexplored habitats. This comprehensive strategy not only enhances the identification of actinomycetes and the bioactive compounds, but it also highlights the unexplored possibilities present in the natural environment and unconventional sources. Now moving to the conclusion. This study, it emphasizes the considerable capacity of natural habitats to severe respiratories for the identifying new actinomycete strains, which possess the ability to produce distinct bioactive chemicals. The results underscore the urgent requirements for the conditions, continuous investigations and improvement of both isolation and dereplication techniques in order to fully enhance the medical potential of actinomycetes. By further developing these methods, we can get more and more new strains of the actinomycetes, which we can use in our further scientific and biological research. Thank you.